My name is Jean Fan, and I'm an assistant professor in the Center for Computational Biology in the Department of Biomedical Engineering at Johns Hopkins University. When we look at a complex organ like the mammalian brain, we may be interested in asking questions like, how are cell types and cell states spatially organized within this tissue? And how does this spatial organization change during healthy development, as well as the progression of disease? By addressing these key biological questions, we may be better able to understand how spatial organization plays a role in tissue function, as well as disease pathogenesis. In order to address these interesting biological questions, it becomes very helpful if we could make precise quantitative measurements regarding the molecular features of cells within the tissue while preserving their spatial organizational information. Recently developed spatially resolved transcriptomics technologies now provide us with an opportunity to begin addressing these interesting biological questions by providing us with the tools to make these large-scaled, high-throughput gene expression measurements of cells or small groups of cells within tissues while preserving their spatial organization. There are many types of spatially resolved transcriptomics technologies. Some, based on spatially resolved capture and sequencing, provide us with full transcriptome untargeted gene expression profiling within small groups of cells within pixels. What these technologies generally allow us to do is take thin tissue sections, such as the one seen here, and allow us to capture the RNAs present at each spatially resolved pixel. By tagging each spatial pixel with a unique barcode and subjecting the resulting libraries to sequencing, we can align the reads to a genome, resolve the original spatial barcodes, and get a quantification of the gene expression at each spatially resolved pixel. Therefore, the process information we end up with comprises the spatial coordinates of the pixels, as well as the gene expression observed at each pixel. As seen here, 2 example genes are shown, and already visually we can begin appreciating how some genes are more highly expressed in some pixels, as noted in red, than others, and different genes will exhibit different spatial patterns due to the underlying cell types and underlying tissue level processes. We can take all of these gene expression measurements and perform clustering analysis to try to identify pixels that are transcriptionally similar to each other, visualized here as a UMAP, or using additional community detection algorithms, cluster them into putative clusters, which we can visualize both on the UMAP as colors, as well as on the original spatial coordinates. In this case, our clustering analysis allows us to identify transcriptionally similar groups of pixels. However, it's worth keeping in mind that each spatially resolved pixel may contain multiple cells, and as a result, these cells may comprise multiple cell types. Therefore, the gene expression measurements we observe may be a reflection of these cell type mixtures. And subsequently, the transcriptionally similar groups of pixels or clusters we identify through our clustering analysis may actually comprise mixtures of cell types rather than one true underlying cell type. And further, the spatial patterns we observe from these transcriptional clusters may also be confounded by cell type mixtures and subsequently hide potentially finer cell type specific spatial organizational patterns. So we sought to develop a computational method that could help us recover these cell type specific spatial organizational patterns and cell type specific gene expression profiles from multi-cellular pixel resolution spatially resolved transcriptomics datasets, all in a way that did not rely on external single cell transcriptomics references. So we developed an approach called STdeconvolve that was recently published in Nature Communications, and you can check out the paper for more details. But briefly, STdeconvolve builds on Latent Dirichlet Allocation, a machine learning model popular in natural language processing. In the context of natural language processing, we can think of each document as a collection of words. Of course, intuitively, these words comprise underlying topics that are latent and not observed. However, we can think of these topics as distributions over words. So by observing many documents, each comprising many words, we can infer the underlying latent topics and therefore estimate the proportion of each topic in each document. By analogy, I hope you can see how each spatially resolved pixel is comprised of a collection of genes. Just as how documents are comprised of underlying latent topics, these spatially resolved pixels are also comprised of underlying latent cell types. And just as topics are made up of distributions of words, we can also think of these cell types as made up of distributions of genes. So by observing many spatially resolved pixels, each with different distributions of genes, we can infer the underlying latent representation of different cell types that must comprise each spatially resolved pixel. In this manner, given multicellular pixel resolution gene expression information, STdeconvolve is able to build on Latent Dirichlet allocation to deconvolve back out the cell type proportional representations in each pixel, as well as the cell type specific gene expression profiles. As a proof of concept, we first sought to simulate multicellular pixel resolution spatially resolved transcriptomics data by taking single cell resolution spatially resolved transcriptomics data and aggregating the gene expression within simulated multicellular pixels. Using this simulation approach, we now know the ground truth cell type proportional representation in each multicellular pixel, as well as the cell type specific gene expression profiles. We can then apply STdeconvolve and evaluate how well we recapitulate both the ground truth cell type proportions and the cell type specific gene expression profiles. And visually, you can see that we do quite well, but we can also quantify this by looking at the correlation between the deconvolved gene expression profiles and the ground truth gene expression profiles, as well as the deconvolved cell type proportional representations and the ground truth proportional representations of cell types across pixels. And in both cases, we see a very high correlation, suggesting that we're able to successfully recapitulate the ground truth. We can also analyze real multicellular pixel resolution spatially resolved transcriptomics datasets, such as this of the mouse olfactory bulb, where we found that clustering analysis alone was unable to recapitulate known subpixel resolution cell type specific spatial organizational patterns, such as the rostral migratory stream in the middle of the granule cell layer, which is not apparent from clustering analysis alone. Applying STdeconvolve, we were able to identify a particular cell type that spatially was localized towards the middle of the granule cell layer where we expected this rostral migratory stream to be present. And when we look into what genes are upregulated in this deconvolved cell type, we indeed find no neuroprogenitor cell markers. And likewise, when we look to see where these genes are expressed using alternative molecular profiling methods such as FISH, we see that they indeed localize to the interior of the granule cell layer as expected. So in this manner, we find that deconvolution with STdeconvolve is able to recover the cell type proportions and cell type specific gene expression profiles of neurons within the rostral migratory stream in the mouse olfactory bulb in a way that clustering analysis was not able to reveal. We're able to apply STdeconvolve to recover cell type specific gene expression profiles and proportions across a wide variety of multicellular pixel resolution spatially resolved transcriptomics datasets and technologies. Returning to our original example, we can apply STdeconvolve to recover the cell type specific gene expression profiles and proportions, and do this for the whole tissue section to reveal interesting new spatial patterns regarding cell types not present through clustering analysis alone. So with that, I hope you've been able to learn a little bit about what is multicellular pixel resolution spatially resolved transcriptomics data, and how we can analyze this type of data with approaches like STdeconvolve to recover cell type specific gene expression profiles and cell type specific spatial organizational patterns. If you're interested in learning more about STdeconvolve, please feel free to check out our lab website, as well as lab blog for additional tutorials.